Okay, welcome to your first real lecture. And this is an introduction to the bacteria, to the prokaryotes. Okay, we'll talk about what they are and a little bit about their structure. You may have had a little bit of this in general bio or something like that, but we're going to go into a lot more detail. Okay. So first of all, let's look at the characteristics. We've already talked about all living cells are made up, all living things are made up of cells. They have a basic shape. They've got a cytoplasm that surrounds it, and they have nucleic acid and ribosomes and those kind of things. You guys should know that already. But you should have learned also that there are two basic cell types, eukaryotes and prokaryotes. Okay? And the eukaryotes, again, membrane-bound organelles that compartmentalize the cytoplasm. They each have a specific function. You guys have learned this. Mitochondria, powerhouse of the cell, blah, blah, blah. Okay? So these are very membrane-bound. They are found in animals, plants, fungi, and protists. And you have this double membrane-bound nucleus with the DNA chromosomes inside that nucleus. Prokaryotic cells, which make up the kingdom of bacteria and archaea, they do not have a membrane-bound nucleus. They have what's called a nucleoid region, but they have no specific membrane-bound nucleus and membrane-bound organelles. They just don't. It's a fairly simple cell. They do have some organelles in there, but they're not membrane-bound type organelles. Okay, they do have ribosomes and things like that we'll talk about, but it's just a different type of cell. Okay, so first of all, this is prokaryotes, very diverse, very numerous. As we talked about in that introduction, they thrive everywhere. They can thrive in a volcanic vent. They can, boiling hot water, hot springs, freezing cold. They can survive, in the atmosphere and clouds. So they can survive everywhere. There's only a few of them that actually are capable of causing diseases to us. I mean, if you think about all the bacteria all over, everywhere, all the prokaryotes, there's only a very few number that actually cause us problems. Okay? So this is what a cell looks like. Okay? And you have a cell wall, which is different from a eukaryotic cell where you talked about cell membranes. Okay? A bacterial cell has a cell wall. It does have a cell membrane inside that. Okay? It may have a capsule on the outside, and we'll talk about that. Or a slime layer may have that. But if you look inside this bacteria, what you see are ribosomes, and you see a nucleoid region, okay? And this nucleoid region is where that nucleic acid is. It is not membrane bound, okay? There may be other stuff in it, and we'll talk about flagellin and membrane and all these wonderful things, but it's a very simple cell. You don't have the Golgi apparatus and all the things that you learned about the eukaryotic cell. It's just not there in the prokaryotic cell. Okay. Now, they do have appendages. I'll go back and look at that. Okay, that allow them to move. Some of them are non-modal, but some of them can move. And they can move using a flagella, which is kind of like a whip-like motion, or they may have attachments that allow them to move. These are fimbriae and pili. We'll talk about that, but they look slightly different. You can see the little fimbriae hanging off. They're like little hairs. And some of them have a surface coating called a glycocalyx. And we'll talk about that in a few minutes. Okay, so first of all, the flagella. Again, it allows them to move. allows them to rotate, allows them to flip over. When we actually look for motility, we look for the fact that the bacteria moves and runs and tumbles and does that sort of thing. And this allows it to move through the environment. If you have an organism that is motile and it gets in your body, it's going to move from one place to the next. And this is how bacteria can invade, because they have this ability to move within the tissue. Okay. So this is what in the lab, in your lab area, I have a discussion on motility, and I have little pictures of these organisms moving underneath the microscope for you so you can see. Okay. But this allows these organisms to migrate into your tissues and go, that's how an infection spreads. Okay. We do classify these bacteria by where the tails are, where the flagella are. Trichus, meaning flagella or tail. Monotrichus, one. It has a single flagellum at one end. Lophotrichus, it has small bunches that are on the same site. Amphitrichus means you have a flagella at both ends, and peritrichus, think of a periscope all the way around, okay? So you can see in the picture, you can see this atrichus without flagella, monotrichus with the one single hair or flagella, amphitrichus, both sides. Okay, we haven't talked about this, but it's basically the same. Peritrichus all the way around, and Lophotrichus, several hairs on one side. Okay, so you do need to note those because that is a characteristic of these organisms. Then you can get the fimbriae. These are very fine hair-like bristles from the surface of the cell, and it allows them to stick. Okay. When you get a bacteria that has these fimbriae, then this bacteria is introduced into the tissue, it can adhere to that tissue and stick, which allows an infection to occur. It's not like you wash it away. So you can see there's the flagella. They are longer, or if you have these little hairs, these projections, the cilia are actually a little smaller than that, but you've got these little projections, all right, like little brushes or bristles coming off that bacteria, okay? And then you have the pili, which is a tubular structure. It's made up of protein, a pili protein. It's found only in gram-negative cells. You don't know what a gram-negative cell is yet, but we'll get there. And its function is to allow for the transfer of nucleic acid. And we're going to get into that, how bacteria exchange nucleic acids, and they exchange it through a process called conjugation. And they use what's called sex pili because they literally are one pili touching another. There's the little fimbriae, the little hair, and you can see, but then you see these structures right here. Those are the pili. So nucleic information, DNA information can be exchanged, or RNA can be exchanged from one organism to another. Okay, and so this is how certain, like if you have a bacteria that, let's say you have a population and you try and kill the bacteria with alcohol, and a few of them survive. Well, those bacteria can exchange that information about how did I not get killed, okay, to other bacteria. And then that makes that whole batch of bacteria that's now growing resistant. Okay? And that happens. And we'll talk about this, but this is how we're going to talk about exchanging resistance plasmids. And this is how these pili can allow for this information about resistance of antibiotics or other chemicals so that the bacteria can survive. Okay. Again, the glycocalyx is a coating on the outside of the cell wall. There are two types. One is called a slime layer, which is very loose, and a capsule, which is very tight. Okay. Slime layers on these, they do protect these. Both of them protect the cells from dehydration and nutrient loss. The interesting thing about a slime layer is it's slippery. So if you have a white cell that's trying to phagocytize this organism with a slime layer, I like to say, think about it. You guys don't do it anymore. But when I was growing up, when we went to a picnic, we would throw a greased watermelon in the pool. And the idea was to go catch this watermelon. Well, as soon as you wrap your hands around that watermelon, it's squirting somewhere else. That's exactly what a slime layer does. It makes it very difficult for a phagocyte to go and get this bacteria because it just slips right through. Okay, so it does inhibit this killing, okay, which makes it more pathogenic. The capsule basically just makes it harder to get into. It really protects the cell from nutrient loss and can allow for the attachment to form a biofilm. And we'll talk about what a biofilm is, but it's where you stick together, and the same thing is like you stick together and you form a mass. Think of when you wake up in the morning, that film that's over your teeth, that's a biofilm. It's formed during your sleep, okay? So just FYI. All right. All right, so let's talk about the envelope, because that's really important in terms of looking at these bacteria and differentiate them. You'll hear people talk about gram-positive, gram-negative bacteria. We're going to get into this, but this is where this is determined, okay? It is the external covering. It is made up of the cell wall and the cell membrane. Again, this is a cell wall, so it's different. It allows the cell to maintain its integrity. There are two groups of bacteria demonstrated, as it says, by the gram stain, and there's a lab on gram stains, so you'll be getting in there. But you have gram positive and gram negative. It is a stain that was invented by Gram that differentiates the difference in these cell walls. The gram-positive bacteria have a different looking cell wall than the gram-negatives. Okay, and we're able to differentiate this, and it becomes very, very important because if you look at trying to treat these bacteria, gram-positive bacteria will be susceptible to different antibiotics than the gram-negative bacteria. So it's very important that when you have a bacteria that is it gram-positive or is it gram-negative? Okay, bacteria don't change their gram stain. Staphylococcus will always, always, always be gram-positive. E. coli will always, always, always be gram-negative. So that it is, whatever it is, that is its staining property, okay? And so we can put these into these categories. All right, so let's look at this. The structure of the cell, as we said, determines the cell shape. It prevents it from lysine, from busting apart. When you have changes in certain environments, osmotic pressure, you go into a different environment, it will prevent this bacteria from, lysine. Like, think of, you should have had, hopefully in general bio, you put a red blood cell that just has a cell membrane and put it in water, it's going to rupture. Okay. You do that to the bacteria? No, It's not going to, because a cell wall is going to protect it. Okay. The main component that makes it a bacteria is peptidoglycan. If you have an organism with a cell wall with peptidoglycan in it, it is a bacteria. If it doesn't have peptidoglycan, then it's and something different. It's archaea. So if you have an organism that is a prokaryotic cell and it has peptidoglycan, it's a bacteria. That's how we classify it. And if it doesn't, it's not a bacteria. It's something else. Okay, so this is a macromolecule, as it says, repeating framework of these sugar chains cross-linked with proteins. And I'll show you that in a second. Okay. And again, it is a three-dimensional lattice work, if you would, okay, that surrounds the cell, and it has alternating sugars, amino amines, and called NAG and NAM. Okay, NAG is N-acetylglucosamine, and NAM is N-acetylmuriatic acid. And they cross-link. And I don't expect you to become a biochemist and go crazy on this, but this is what it looks like. You get this lattice work, and they're just tied together, NAG, NAM, NAG, NAM, NAG, NAM. And it makes this cross-hatch lattice work around the cell. Okay? And you can take a look at these two different biochemicals, but I don't expect you to memorize it, so don't go crazy. But it forms a lattice work around that cell. Okay? It's pretty thick. It's in a gram positive. Okay, and here's the difference between the gram positive and gram negatives. Gram positive has a thick peptidoglycan layer. Okay, you can see it's 20 to 80 nanometers, which is tiny, but you're talking about a tiny cell. And it includes teichoic acid and lipoteichoic acid, and it functions to maintain that cell during cell division. So it's actually going to bud or go through binary fission, and it allows that cell wall to be maintained and to enlarge when it's going through cell division. Okay. Some cells have a space between the membrane and the cell wall. If it does have one, it's a periplasmic space. Not that important in terms of what I want you to know. What I want you to understand is gram positive, that cell wall is a thick cell wall. Okay. Gram negatives. So here's your gram positive. Okay. You have this very thick, you can see this teichoic acid hanging out there. But here's that peptidoglycan layer. And then you have a space, this periplasmic space. And then you have the cell membrane. We'll talk about that, but that's where your cell membrane is. But look at the thickness of this layer of peptidoglycan. And this becomes important. Okay, and you can see how thick it is. I don't expect you to do the biochemistry. Okay, but I expect you to know the thickness. So you have a thick peptidoglycan layer, you have a space, and then you have the membrane. This becomes important because when we start doing the gram stain. Okay? Gram negatives have an inner and outer membrane with a space between them that contains a thin peptidoglycan layer. Okay? Thin. This is different. The outer membrane is called the LPS because it contains lipopolysaccharides. There is a lipid portion of it. It is a toxin. It is an endotoxin. We'll talk later about endotoxins, but it makes these gram negatives a little more pathogenic, and they can become toxic when they are released during infections. Okay. And they have porn proteins in that upper layer, and they regulate what enters and leaves the cell. So you take a look at the gram-negative. Well, this looks a whole lot different. Okay, first of all, look at your peptidoglycan layer. It's thin. It's thin right here. It's very thin compared to this puppy. Okay, this is your outer membrane. This is your polysaccharide. This is where your toxin is, right out here, this layer, right? Then you have your space and you've got your cell membrane inside. So you can see this, all right? So this is a totally different structure. And again, learn the basics. And then when we talk about the gram stain, it'll become very clear because we can wash this off, and this is how we can actually stain it, because this layer is so much thinner than this layer, okay? But that is the difference between the two. So gram-positives, thick peptidoglycan layer, no LPS. You go from the peptidoglycan layer into your membrane, okay? Gram-negatives, you have an LPS, lipopolysaccharide outer membrane, you have a space, you have a thin peptidoglycan layer, and then you have your membrane. Okay, that's the big difference. So if you look at the two, here's the comparison. Number of layers, 1 in the gram-positive, 2 in the gram-negative. You can look at the chemical composition, but you can see the teichoic acids are not in the gram-negatives. The LPS is not in the gram-positives, okay? The overall thickness is completely different. Look at that, much thinner. Gram-positives do not have an outer membrane. Gram-negatives have that outer layer, LPS. The space in the gram-positives is narrow, whereas the space in the gram-negatives is pretty extensive. So gram-positives are more permeable to molecules in terms of you can get stuff in, whereas gram-negatives are not because of all this layering that has. Okay. Let's get into this cell membrane. Okay, and it is a phospholipid bilayer with proteins and provides the site for energy reactions just like we have for our cells. It is a passage for nutrients into the cell. It's a way to get rid of waste, and it is selectively permeable just like a eukaryotic cell. Okay. All right, so if we look at this is the cell membrane, and you can see there are transport proteins, there are binding sites, there are phospholipids, there are glycolipids. This cell membrane acts kind of like the same cell membrane that we have in our cells. Okay. So inside the bacterial cell, it is a gelatinous solution of sugars, amino acids, salts, mainly water, and it's a solvent for the materials used in the cell function of the cell. All right, a lot more stuff inside our cells. All right, then we get into this nucleoloid region. Again, we don't have a membrane-bound nucleus. Humans have 46 chromosomes. Bacteria have 1. Okay, a single circular double-stranded DNA molecule. So when you sit there and look at this chromosome, you look at all our chromosomes, they look like little X's and stuff like that. They're not circular at all. They are double-stranded, but we got 46 copies. 1 chromosome. It is circular. It is double-stranded. It contains all the genetic information required by that bacterial cell. Now, in addition to that, floating in the cell are these small circular pieces of double-stranded DNA that we call plasmids. They do not contain information about bacterial growth or metabolism or anything else, but they can be readily manipulated and transferred. And we'll talk about different kinds of plasmids. For example, resistance plasmids that we just talked about and getting resistance. These bacteria can exchange resistance plasmids from one bacteria to another. And we'll talk about that a little bit later on. But realize that you got one circular little chromosome and that's it. Okay, the ribosomes of a bacteria are smaller than our ribosomes. Okay, they have 2 subunits, and prokaryotic cells differ from eukaryotic cells in the number of proteins and in the size. But ribosomes in the bacteria are the site of protein synthesis, just like the ribosomes in the eukaryotic cells. Okay, I've got a little picture here, and you can see the difference. Here's the eukaryotic ribosome, and we say it has a 60S subunit and a 40S subunit, and we add that together, kind of weird, and get an 80S. The prokaryotes are slightly smaller. They have a 50S and a 30S unit, so it is a smaller ribosome. Okay. All right. The other thing that they can make, which becomes very important, is an endospore. And it's produced by some gram-positive genera, particularly Clostridium and Bacillus. Okay, and these are inert resting cells, and they have 2 phases: the vegetative state, where they're active and they're growing, and when there's adverse conditions occur, it moves its genetic material into this endospore, and it can survive all kinds of different environmental conditions and very long-term survival. And this becomes important when you think about, go back here. This becomes important when you think about these organisms, because Clostridium botulinum causes botulism. Bacillus causes the same thing. Clostridium tetani causes tetanus. So this is how, like, a rusty nail can have this bacteria sit on it forever. You step on it and you can get tetanus. Just FYI. Okay. So when we'll talk about these organisms later on. We'll get into a lot more specifics, believe me. Okay. But understand that these organisms that form endospore can last out there for years. And this is why if you step on a rusty nail and you haven't had your tetanus shot, and in certain areas, I would say in the States, it's usually 10 years. If you go travel certain places, the recommendation is that you have a tetanus shot within 3 years. Okay, so go check that out. So, and this is because Clostridium has this endospore, and when it's in a dry condition, it's just sitting in there, happy little thing. And the second the conditions get better, that Clostridium start reproducing. Okay, so very long-term survival. We'll talk about these a little bit later. Sporulation is the formation of these endospores, And again, it's the hardiest of all our life forms. It can withstand heat, for you, effect of UV light, and you're going to see how long it takes. We're going to expose UV light to staph and UV light to bacillus, and you're going to see how much longer it takes to kill the bacillus because of that endospore. Okay. It's not a means of reproduction. It is not how it reproduces. It just allows that organism to survive. Okay. And then when it gets into a better conditions, it germinates and allows it to reproduce and grow. Okay. So again, you see longevity versus immortality. Years. It is metabolically inactive. You have to boil it for a long time. Even pressurized steam, you got to go for 20, 30 minutes to kill it. UV light, you got to go at least direct exposure for at least 10 minutes. So this is why when you start talking about trying to sterilize it, pasteurization, trying to protect your foods, you have to think about this stuff because this is what you're trying to kill. Okay, just FYI. This is what an endospore looks like. This is actually an endospore stain, and we'll take a look at that. But you can see the bacillus. There's that little endospore in there. Okay. All right. Prokaryotes reproduce asexually. All of them reproduce asexually. They reproduce by binary fission, which is the most common. They can do snapping division. They can have reproductive spores. They can bud, all kinds of stuff. All right. So let's kind of look at these. Binary fission is just, as it says, it's dividing in two. Binary fission. You have a parent cell. The DNA replicates, the cytoplasm divides, and you end up with what we call two daughter cells. And those two daughter cells divide, and we have now 4 cells. And this is binary fission, and this occurs in a bacteria about every 20 minutes. So every 20 minutes, that bacteria, when it's actively growing, is dividing and doubling itself, which is why you can get millions of bacteria in just a few hours. Okay, so think about that. When you get a bacteria introduced into the tissue and it's viable and dividing, you can have an infection pretty quickly. Okay. You can have snapping division, where it just basically makes a V snap. This is kind of unusual. I've seen it got this certain bacteria are identified. We'll talk about this when we start to go through these organisms, but they are identified by this V snapping shape. Okay. Cholera is one of them. Does this. And you can produce spores. Bacteria can produce spores. We'll talk about the prokaryotes and their spores. One that's very common is budding. Binary fission is primary, but you can get budding. Yeast will bud. Fungi and stuff like that will produce spores. But it's notorious for yeast to bud. And you form this little young bud, and then you have a daughter cell. And when you see a report, let's say you get a urine analysis and you see a report that says budding yeast seen. What that means is there's an active yeast infection because that yeast is actively budding. Okay. So we'll talk about that a little bit later on. Okay. Now, arrangement of prokaryotic cells. This is important too. This is when you do binary fission, prokaryotes divide in different planes. And with this, some of them separate individually and some of them form chains. This becomes very important because, again, just like the gram stain, there are certain ways that certain species always divide. And we'll talk about this in a second. But if I see gram-positive cocci in clusters, you'll understand that in a second. That's a staph. If I see gram-positive, bacilli in chains, it's something else. If I see gram, it's a strep. If I see gram-negative bacilli, we'll talk about this. But these bacteria always divide the same way. Okay? And we'll talk about this in a second. Okay, so the different shapes, there's 3 basic shapes: cocci, bacilli, and spirillum. Cocci are round, spherical. Bacillus is rods. Now, you can have very short rods, which are called coccobacillus, or they can be a little curved, which is vibrio, that's found in cholera. But we'll look at this in a second, and there's a lab on this, okay, to make sure you go through this, and I'm gonna have a lot of pictures of these, and I'm gonna have you identify them, so don't panic. And then you have spirillum, which is actually a helix. It's a rod, and the spirochetes are kind of like looking like a spring. So when you look at this, there's our little cocci, two little dots together. There's the bacillus, which is a rod shape. The vibrio is a rod, but it's curved. And then you have these spirillum and spirochetes, and then you have some really odd ones out there that we're not going to go over, but there are some odd ones. There are changeable ones. Okay, so here's the key. Cocci can divide in different planes. Okay? There are cocci that divide only in one plane. And that is if it forms 2 cocci together, it is called a diplococcus. Okay? And there are certain diplococci that we look for. We'll talk about that as we get through these organisms. If it continues on and forms a chain, it is called a streptococcus. Okay? We'll talk about those. Strep throat. Okay? That when we get a throat culture and we see this is the kind of stuff that we're looking for. We're looking for those chains of cocci, right? It can divide in 2 or more planes. If it's in 2 planes, it forms a tetrad. 3 planes, sarcine. If it divides in random planes, just keeps dividing, it's a staphylococcus. So keep in mind, some of these will break apart, and we'll take a look at this under the slides. But if you have a staph, a lot of times you may see it reported as in the smear. Cocci seen singly, in pairs, and clusters. Okay? That means when we looked at the smear, we saw this cocci just beginning to divide, and then it divided again, and then it finally clustered around, and that's a staph. That tells us it's a staph. Okay? So these shapes and these arrangements give us a real good clue as to what the organism is. So when that doctor orders a gram stain, I need a stat gram stain. I have somebody that I suspect has a bacterial infection, maybe. And so I'm going to do a spinal fluid tap, and I want to see, and they're going to order a stat gram stain to see if they have a bacterial infection that's in their spinal cord. Not a good thing. And we're going to do it, and we're going to do a gram stain on it and report it, because that will tell the doctor, give them an idea of what to treat with, because certain antibiotics will treat gram-positive, certain antibiotics will treat gram-negatives, some antibiotics are better on staphs, streps, etc. And we'll talk about that when we get to the chapter on antibiotics, because we'll talk about why. But at this point, understand that you have the shapes and the way they divide. Okay, and again, I'll have a lab on this, but let's, understand. Bacilli only divide on one plane. They cannot do this one plane, two planes, multiple planes to form clusters. You will never see bacilli in a cluster, ever. Okay? You may see a single bacillus. You may see a chain. Remember what we call the chain of cocci? Go back. What's that chain of cocci? Streptococcus. Strep means chain. So if you see a chain of bacilli, it's a streptobacillus. Okay? It may palisade, form those V's that we talked about. Okay? And it may be in two. Remember diplococci? Here's diplobacilli. And again, these arrangements are characteristic of these organisms. And this is what we look for when we're trying to identify these organisms. If we only see diplobacilli and we don't see any chains, it's just not there. Okay, it's not an E. coli. Okay, and this will make more sense as we go through. All right, so how do we classify these guys? As we talked about, we classify them. We used to have a hard time classifying them because we didn't know where they belong, but now we classify them according to their ribosomal ribonucleic acid, this rRNA, and put them into these three domains, either a eukaryote, bacteria, archaea. Right? So we start classifying these bacteria into what group they belong in. If we find something, we start using a ribosomal RNA to do this. When we look at archaea, I'm just going to go through this briefly. They lack peptidoglycan, as again, the differential between archaea and bacteria. Bacteria has peptidoglycan, archaea does not, has something else. Okay. It does reproduce similarly, but binary fission. It can bud. There are cocci, there are bacilli, there are spirals, they're pleomorphic. So they can look like a bacteria, but they're not. They are not known to cause disease because primarily they are extremophiles. They live in these environments that, we just couldn't live in. This is what they look like. You can see that pleomorphic right there. Okay. So they are extremophiles. They exist in extreme conditions, extreme temperatures, hot springs, extreme pH. They get in acids, strong acids. This Great Salt Lake in Utah, that high salinity. Okay, they are thermophile. We'll talk about this. That makes them a thermophile or a halophile. Those high salts. So they are prominent members in these areas. The thermophiles, here's the interesting thing is their proteins, their nucleic acids do not function below 45 degrees Celsius. Body temperature is 37. So they don't function below about 105, 106. And then you have the hypothermophiles, which require temperatures to be over 80 Celsius. So this is getting really hot. So again, you're talking about what gives the color to, I spelled that wrong. What gives the color to our hot springs? Okay. I'll correct this right now. What gives the color to our hot springs in Yellowstone? It's these hyperthermophilic members of archaea. Okay. All right, your extremophiles, again, we use them in research. We use them in, to replicate DNA. We use them in biotechnology because trying to break apart these sequences, we need an enzyme that will last at high temperatures. And we found it in bacteria. So you ever take a course in biotechnology, you will learn about that. But yeah, we use a specific enzyme to do this, and it is a thermophile. Okay. And again, the halophiles, you have to have greater than 9% salt. So, and they may have pigments in them. The most studied one is this halobacterium. And again, this is Great Salt Lake. You can see the different colors, highly saline water. But again, these guys don't cause us problems. Okay. We can't close this out without mentioning phthalagens. This is the largest group of archaea. They convert carbon dioxide, hydrogen gas, organic material to methane. Primary source of environmental methane, a lot of it is buried in our ocean floor. If that ever broke apart, we'd be in deep trouble. But it digests the sludge during sewage treatment and converts organic waste to methane. And if anybody ever been in a swamp, you know what we're talking about. You know, that's swamp gas, that's methane. Okay. So that is our introduction to prokaryotes. We'll get more into this. I kind of went into it a little bit. We're going to get deeper and deeper. But again, with the gram stains, it's going to be in the lab. We're going to work through that so you guys can remember that. Okay.